everybody, welcome back to my channel Pharma as well. This is Devi Sarojini. The main purpose of this channel is simply answering all your queries related to pharma. If you are related to pharma or if you have any query related to pharma, please feel free to leave a comment, like the video, and share to your friends, and don't forget to subscribe. Now I will discuss about the comparison and differences between two major guidelines about OOS, they are MHRA and USFDA. This is definitely something to discuss. Also, this video gives you a lot of information and knowledge about two major guidelines about OOS. Watch the entire video without skipping. If you are watching this video for the first time, be sure to like, share to your friends, and don't forget to subscribe. Now coming to the definition of OAS. What is OAS? OAS means out of specification, isn't it? Is it enough for the definition? The clear definition for the OAS, like this: OAS refers to a product that does not meet the specifications defined for it. The product that does not meet the specification defined for it. This indicates a deviation and could have occurred at any stage of the product. Uh, Needing a thorough investigation. The OOS result could also be a laboratory based error. In some cases, it could also be a laboratory based error. This should be eliminated before deciding to reject or retain a batch. I will explain it clearly. During the course of a drug development and as the product is nearing completion, it needs to be tested to ensure that it performs as expected within the specified limits. These specified limits are mentioned in the compendia. The product needs to be tested to ensure that it performs as expected within the specified limits. When it falls outside the specified limits, then it is said to be OOS. When it falls outside the established acceptance criteria, then it is called OOS. Now I hope you understand it. If this happens often, it is an indication of the manufacturing and analytical procedures not being in control. It can lead to frequent customer complaints and the rejection of commercial batches. As a result, the pharmaceutical business will have a heavy inventory loss. It may also compromise the safety of patients and their handlers. Therefore, the incident of OS result occurrence must be investigated and the root cause addressed. From the time of design to the time of manufacturing, the design undergoes several changes. Due to these changes or due to process errors, deviations may be introduced at the time of manufacturing too. This can cause OOS. Some of the common factors for OOS: deviations. Deviations during the product manufacturing process, errors during the testing due to incorrect procedure, errors caused by malfunctioning analytical equipment. These are the common factors for the OOS. Therefore, to understand whether the error is due to the product not conforming to the specification or other factor, a root cause analysis must be performed to identify the true cause. Regulatory agencies around the world have guidelines that provide a framework for the investigation and handling of these results to ensure product quality and patient safety. Uh, Two major regulatory agencies that provide guidance on OS results are the US Food and Drug Administration and the UK's Medicines and Healthcare Products Regulatory Agents, that is FDA and MHRA. Here a small comparison between the FDA and MHRA guideline on OS handling. First one, US FDA guideline was published in October 2006 and lastly updated in May 2020. Recently, MHRA published in August 2013 and it was lastly updated in February 2018. Definition for OS, according to USFDA, OS results are those that fall outside established acceptance criteria, which have been set in official compendia, drug applications, or by the manufacturer. MHRA. Similarly, MHRA defines OS results as those outside of specified limits set in the marketing authorization are defined by the manufacturer. Here we are noticed that both US FDA and MHRA give same definition for OOS. Next one, initial assessment. Once an OS result is identified, a preliminary assessment should be conducted. Preliminary assessment means initial assessment. It should be conducted to determine if a clear and obvious error has occurred. If an error is found, the result can be invalidated. MHRA also recommends an initial assessment to check for obvious laboratory errors. Next one, phase one investigation. Both FDA and MHRA both focuses on phase one investigation. This phase often focuses on the laboratory procedures and processes. In FDA, if the result cannot be invalidated through the initial assessment, a full-scale investigation is initiated. This phase often focuses on the laboratory procedures and processes. MHRA. MHRA also similar approach with a recommendation for a thorough investigation, starting with the laboratory where the OS result was observed. On observing an OS result, a laboratory preliminary investigation is recommended to identify the assignable cause. Here we are noticing that to identify the assignable laboratory procedures to identify the assignable cause. But what is assignable cause? Assignable cause means when the error is identified, it is called assignable cause. When the error is not identified, it is called non-assignable. Based on the findings, further investigations will be conducted. Next one, phase two investigation. If the OS cannot be attributed to laboratory error, the investigation expands to areas outside the lab. Areas outside the lab means manufacturing processes, raw materials, and other areas that could impact the result. MHRA also focusing on the manufacturing process. That is, the investigation expands to outside the lab, focusing on the manufacturing processes. Both FDA and MHRA, the investigation expands to outside the lab, focusing on manufacturing processes. Next, retesting. FDA allows retesting to confirm the OS result. The number of retests should be predefined and justified. MHRA also permits retesting. However, simply obtaining a passing result in a retest does not negate the original OS result. Both FDA and MHRA permits retesting OS result, confirming the OS result. Next, documentation. FDA emphasizes the importance of documenting every step of the OS investigation, including all discussions, findings, conclusions, and follow-up actions. MHRA. Similarly, MHRA also stresses the importance of thorough documentation. Next one, quality system. FDA emphasizes that OS results should be handled with a framework of pharmaceutical quality system, which may trigger corrective and preventive action. MHRA also recommends integrating OS handling within the quality system of the organization. Last one, conclusion, that is final assessment. FDA and MHRA both requires a conclusion based on the evidence gathered during the investigation. If the cause of OS is identified and corrected, the batch may still be released. If not, the batch may be rejected. In MHRA, a conclusion should be drawn based on the investigation findings. Corrective action should be determined and batches may need to be rejected if no cause is found. Here, all this about comparison between MHRA and USFDA guidelines about OS. Now, I will discuss about the differences between MHRA and USFDA guidelines about OS one by one. First one, investigation phases. There are two phases in USFDA guideline. Investigation phases. They are phase one and phase two. MHRA guideline divides OS investigation phases into three phases. Those are phase 1, phase 2, and phase 3. In USFDA OS guideline, there is no further subdivision of phase 1. But in case of MHRA, we are noticing that phase 1 again divided into two phases. They are phase 1A and phase 1B. USFDA guideline talks about OS outlier result investigation. However, MHRA guideline talks about OOS, poverty, outlier, and atypical results investigation. Next one, microbiological investigation. USFDA guideline does not talk about microbiological investigation. In MHRA guideline, we will find that microbiological investigations are precisely elaborated. Next one, stability. OOS, poverty investigations of stability sampling not covered in USFDA guideline. In MHRA guideline, we will find that stability samples, OOS, poverty investigations are covered separately. Next one, USFDA guideline does not talk about 95% of confidence limit concept while evaluating the mean value of the retesting results. But in case of MHRA guideline, 95% confidence limit concept should comply. Uh, Next one, no quality assurance term is used in USFDA guideline. MHRA guideline, we will find that the term quality assurance has been used throughout the document. Here, all is about the differences between MHRA and USFDA guideline. I hope you understand. Thank you for watching. Thank you, guys.